Welcome to the lecture on DNA transcription, which is the synthesis of RNA from the DNA template. In our learning objectives, as always, a lot of detail in this lecture, and it's just important to remind you that the lecture objectives will tell you what it is you need to know, and then I always say to the level of detail presented on the slides will help you know how far to go. In general, what I mean is, if you're able to take any slide and explain the picture that you see by telling the story, you'll be in good shape. So transcription now is going to be the way that the genetic information contained within a cell is turned into something that is actionable. So the DNA is like the blueprint for proteins and processes and life and all that kind of stuff. But we need to make a copy of that DNA that can go out into the cell and make things happen. And so you're going to transcribe the DNA message. You're going to transcribe it into some form that's able to be acted upon. Now, DNA can be acted on. Of course it can, of course. But the real workhorse is going to be the RNA in the cell, which is the primary transcript of the DNA. So the genetic blueprint is the DNA sequence, and then the RNA becomes the working copy of the DNA. Now we know that RNA, ribonucleic acid, is not the same as deoxyribonucleic acid because of the difference at 2' OH and H and the bases and stuff, but the RNA is what goes out in the cell and does the work. Now the copying process, making the RNA copy of the DNA, is what is the process of transcription here. So here's your DNA genetic blueprint, and the process of transcription will produce RNA, and the RNA will go out in the cell and do things. It could be a transfer RNA, which is responsible for protein synthesis. Here's ribosomal RNA, which is the machine that helps synthesize those proteins. There is the messenger RNA that carries the gene information about what that protein is supposed to be here. Now recognize that the transcription process is highly selective, okay, which means the cell will know exactly what gene it wants to transcribe, how much of that gene to transcribe, how long to transcribe that gene. It's very, very regulated in terms of you only want to transcribe the DNA if it's time to transcribe it. And there'll be specific signals that will tell you to do this. And we have a lecture coming up and a couple more lectures about just that. And there's going to be a lot of post-transcriptional modifications in the eukaryotic cells, where you can convert the primary transcript, which means if you make a copy of the DNA, whatever comes off is going to be in its raw, its unedited form. And in eukaryotic cells, you're going to modify that primary transcript into something that's a final form that is really highly fine-tuned for what the function is going to be. Let's compare eukaryotic and prokaryotic transcription processes. Here, prokaryotic on the left, eukaryotic on the right. And of course, prokaryotic is going to be a little bit simpler. The similarities between these two cell types is there's the DNA, which is the genetic code. And you're going to use the transcription process to make the copy, the RNA copy of that DNA genetic code here. And off will come an RNA product here and there. And for the prokaryote, it's much simpler. The DNA will have a code on it that will lead to the production of an mRNA. And this mRNA is really pretty much, it's this final form. As it's coming off the DNA, the bacteria recognizes that messenger RNA and will immediately start to work on that messenger RNA to accomplish protein synthesis. And the regulation now is going to be very complex within the messenger RNA as it comes off of the DNA. But you see, very simple. DNA, primary transcript, work of the cell. Eukaryotes, of course, is a little bit more complicated. Again, here's the genetic material. Here is the pre-mRNA. And this molecule is going to be very large compared to what the molecule needs to be. There's going to be in this molecule all the stuff that's required to go out in the cell and do what needs to be done. But there's extra sequence in here that's important for regulation and for preparing the final copy of the mRNA. There's different ways that this thing can be cut and recombined. It's called splicing. And by doing different ways of splicing, you can make very similar proteins that are slightly different, have slightly different function. But all of that information is contained within this pre-mRNA sequence. And so you have what's called the RNA processing step, where these things are done to make this final piece of mRNA that is fully functional and ready to go. And at that point, the RNA leaves the nucleus, goes out in the cytoplasm where it can go to work. And in this case, you're showing a messenger RNA, which is going to be picked up by a ribosome, and you'll have a formation of a protein out here. So the overall flow is similar, but the details for the eukaryote, it's a little bit more complex. We had DNA polymerases when we wanted to synthesize or copy the DNA in a cell, and now we're going to have RNA polymerases when we want to make a piece of RNA using the DNA as the instruction piece for how to do that. So let's look at how these enzymes work, and then we can talk about differences between prokaryotic and eukaryotic. So here's the RNA polymerase. It's an enzyme, and as an enzyme, it's got an active site, and that active site is where the chemistry of the chemical reaction will take place. And like any enzyme in the active site, it's going to have a very specific structure that will recognize the DNA to which it will bind, biomolecular recognition. The design of the inside of this active site is perfect for a thing called the start site or the initiation site on this piece of DNA. So here's the DNA, 5' to 3', it's double helical, so you've got a 5' to 3' and an anti-parallel thing. And this little bit here, this little bit of sequence now will be recognized by the RNA polymerase. And that is where the polymerase will bind, and it knows to start synthesizing the RNA at this position, and it will check it all the way through this DNA sequence until it comes to something called the stop site. Again, this is a couple of DNA base pairs that the RNA polymerase will recognize, and when that sequence comes up, it causes the RNA polymerase to stop and to come off of the DNA. So you end up only making an RNA transcript that matches the DNA between the green and the red bulbs here. So the RNA polymerase is kind of interesting. For the DNA, we had to have a helicase and a topoisomerase and the polymerase. The RNA polymerase does it all by itself. It is responsible for unwinding the double helical DNA, and it takes one of the strands and just sort of sets it aside, and it's going to operate on only one of the two strands here. Remember, the DNA polymerase's job was to copy both strands to make an exact copy of the double helical DNA. For the RNA polymerase, what you're doing is you're saying, I want to make a copy of this genetic sequence and this bit of DNA that's going to tell me exactly what I need to know about making a protein. So we call this melting. Melting means when you take two things that are held together and you pull them apart or you have them come apart, it's called melting. And, you know, if you have any laboratory experience with DNA, you know, you can heat DNA up and it melts, but that just means taking it apart. So once it becomes apart now, that allows this RNA polymerase to operate on one strand and one strand only. And as we saw before, you're going to have a polynucleotide synthesis reaction taking place, where it's very hard to see there, but that little tiny bit of red, right there to the right of that green ball, is the initial ribonuclear nucleotide triphosphates. And so the R means that you're looking at the oxy form. So the 2 position has that OH on it. And the RNA polymerase has the ability to start synthesizing immediately. It does not need a primer the way the DNA polymerase does. It can start right at position number 1 and immediately base pair with the RNA bases and start making a copy of that DNA strand. And sure enough, chugga, as this RNA polymerase moves to the right here and continues to copy the DNA strand, you see what's called the nascent. Nascent means newly formed. And the RNA strand is coming out here. Eventually, this RNA polymerase will reach this termination site here, and it says, hey, I need to stop. It stops the synthesis reaction. It lets go of the RNA transcript here, and it comes off of the DNA. And in the process, you see that as the RNA polymerase is moving along, that DNA double helix is reforming. And so as the RNA polymerase comes off, the double helix is reestablished. So the DNA is just fine. And then here is your completed RNA strand. It's called the transcript, and the polymerase is now free. So a lot of words here, but this is the same logic as what we had before, and I really do like this picture. I wish there was one like it in the DNA lecture. But as you'll notice, on the right here is the DNA template. What is a template? A template is going to be that structure that you're going to want to copy. It's going to be the instructions on what you need to add next. And how are those instructions conveyed? Well, we know that these are nitrogenous bases. They are nucleotides. They've got these base structures here. And we know due to base pairing that for RNA, you'll have the U and the A will base pair. And you'll know that the G and the C will base pair. And RNA is going to have A still. And so if there's a T on a DNA, that A can still bind to the T on the DNA, even though the RNA doesn't have any T's. And this U, of course, because normally T and A base pair, you have this U in place of the T. So the instructions are that these two bits of that polynucleotide will bind together by the normal base pairing. And to extend this by one more base, just like we saw before, a new nucleotide triphosphate molecule will come in and will base pair, in this case U and A base pair, U because it's RNA, and they will form that base pair interaction, which will position the 5' first phosphate right beside that 3' OH, which will allow that 3' OH to attack that phosphate, form the phosphodiester linkage. It removes the inorganic phosphate, the PPI, the two phosphates there, and that will connect this new, in this case, a UTP molecule on there. So you're now one base longer. And again, you're going from the 5' to the 3' direction. The chain is getting longer. So the template is telling you how to do that. So what would come next? If you have a T here, what does T normally bind to? Well, A. So an A base will come. And then A, for RNA, what would come next? U, right? Because there's no T in RNA, it's U instead. And then we'll have a C. So it's like we saw before. And so the RNA polymerase's job is just basically to run down this chain and add. And what this note says here is that the RNA polymerase doesn't have exonuclease activity, although they can do some proofreading along the way there. So again, you're going to have fairly faithful reproduction here, but the DNA polymerase will add the next DNA base, and it will do a quick check to make sure the base pairing is correct. And if it's not correct, the DNA polymerase will pull that base off and allow the next base to come in that's correct and then put it on there. And the RNA polymerase doesn't really do that so well. Okay, and here's the overall error rate, 1 in 100,000 bases. So you see the error rate is greater for the RNA polymerase because it doesn't have that ability that the DNA polymerase has. And yet there is error checking, and we'll see. So there's a couple types of RNA polymerases, and it depends, of course, on whether you're eukaryotic or whether you're prokaryotic. And again, the prokaryotic system is going to be very simple. The prokaryotic cells have a single RNA polymerase. So when we're talking about the prokaryotic cells, we're just simply saying the polymerase, the polymerase, the polymerase. Okay. Now, there are several subunits here, and we're getting a little bit into the weeds here, but there are subunits that form the core enzyme, and of course those subunits are going to have recognition and work and chemical, and all these things work together. But key here is what's called the sigma factor. The sigma factor is absolutely essential for this RNA polymerase to bind to DNA, to recognize its initiation site, and for that RNA polymerase to be activated so it can start doing the transcription activity. The sigma factor must be there. It's going to bind to this core enzyme complex, and it's going to direct the RNA polymerase to that promoter sequence. That where do we start sequence is what the promoter sequence is. We have a nice picture coming up here. Okay. So there's several different sigma factors. Okay, you'll see there's actually a bunch, and that's more detail than we really need to go into. But the major factor, the one that is really the fundamental factor, is going to be the sigma 70. So you'll see that. Okay. Now in eukaryotes we have 3 different RNA polymerases, and they're listed over here, and it just so happens that they work with the 3 different types of RNA that we've known before. RNA polymerase 1 makes ribosomal RNA, RNA polymerase 2 makes messenger RNA, and also these things called small microRNAs, which are involved with regulation of messenger RNAs, and you'll see more of that. And then RNA polymerase 3, of course, does the tRNAs. I've used the term promoter several times, but now it's time to look at what a promoter is and how it works. And we'll do the prokaryotic promoters because they're a little simpler and a little easier to understand, I think. Now the main issue is this. Here's this long strand of double helical DNA. And within that double helical DNA, there's going to be genes, and those are the genes that need to be turned into RNA that can go out and do the work of the cell. So the RNA polymerase has to know where those genes are. How in the world does it know in all of this DNA to bind to the exact spot where it's supposed to start transcribing that gene? Well, remember the sigma factor, and there are other factors that will bind, but sigma 70 will bind to the RNA polymerase, and together they will be able to recognize very specific sequences on the DNA strand. If you go down, here's a whole bunch of DNA strands from a bunch of different bacteria, prokaryotic cells down here. And over the years, it became very clear that there were these consensus sequences. Some places here where the DNA sequence is exactly identical. Exactly, just so close. If it's not exactly identical, it's really close, And then over here, you see some common features as well, although this doesn't seem to be quite nearly as identical. And over the years, it was determined that these represent very distinct binding sites or interaction sites between the machinery of RNA transcription and getting things positioned on the DNA properly so that they can find and recognize these initiation sites and then transcribe the genes that go to the right from here. And so the promoter now is going to be this region of regulatory sequences that is going to control the binding of the RNA polymerase to the DNA, and it's also going to help the RNA polymerase to know where that initiation of transcription or that start point is. And there's going to be a variety of proteins and factors that bind and help everything get positioned just right. And so this whole thing works together to get everybody positioned just right and everything in place so that transcription can start. So this is a really nice picture, and I like this. And this is showing you how the transcription process is working. And again, let's start with our double-stranded DNA. And this is showing you the RNA polymerase is already doing the work. It's already melted or separated these two strands of the DNA, and then you'll notice that it's copying this strand down here. And if you recall, this is the base pairing, and that's helping these things stick together. But where the new synthesis is taking place is up here. And that's where you're getting the initial base formation, you're getting the elongation, 5' to 3'. It's getting longer as you move to the right. And so the direction of transcription shown by the arrow is you're moving, and from this picture, you're moving from the left to the right. And that is allowing you to read this DNA. Now, there are two terms you've got to see here, DNA coding strand and DNA template strand. So the DNA coding strand, that is the DNA that contains the code. That's it. That is the information. That's the real thing. And that's what we need to have out in our cell. But if you were to make a copy of the coding strand, you would actually end up making a complementary copy that would be opposite of the bases of what you need. And that would send information out that would be the opposite of what you need from the coding strand. But as you know, the DNA double helix is going to have a coding strand, and it's going to have what's called a template strand. And if you take that opposite strand that is perfect for the coding strand in the DNA double helix, if you copy the template, you will reproduce the sequence that is identical to the coding strand. Of course, you'll use the RNA bases. You'll use the U instead of the T, and you'll have the 3' OH and stuff instead of, sorry, 2' OH instead of the whatever, but the sequence here of this RNA will match exactly the sequence of the coding strand. And that is why we do the template strand and not the coding strand up here. Now the eukaryotes also have promoters, and you're going to see quite a bit more complexity here. And I didn't mention it before, but there's some cute names. There was the Pribnow box and the Tata box, and these things that are consensus sequences that are recognized by the transcription machinery. And so eukaryotic cells just have more of these things. If you recall, over in the prokaryotic promoter, there was 2 areas of consensus, and then there was an initiation site. So quite a bit more here. And you see, compared to the start site where these things are supposed to start, this is showing you approximately how many base pairs you're going to have and about where it might be located here. So for example, if the transcription site is here, then you're going to have these consensus sequence somewhere down the line here. Sorry, I can't speak. Consensus sequence somewhere down the line. And again, these sequences are going to be responsible for very specific factors that will bind. By the word factor, we mean proteins that will bind. They will recognize these sites, bind there, and then they will help recruit other proteins that are necessary for the transcription process. So this is a really nice picture again here, how these factors work together. So here's the Tata box, and the Tata box is going to be that primary site that says, let's start here, let's bind to this little consensus sequence of DNA. And what you find is all these proteins are listed off over here. So the Tata binding protein, the TBP here, recognizes that sequence and it binds. And once it binds to the DNA, just like everything else, it goes through a conformational change, and that makes it attractive for other proteins to bind to it. And so you see a series of coactivators. These are not named, but there's all kinds of them that are out there. There's transcription factors here, TF, they all have numbers. Transcription factors over here with letters, okay? And then all these transcription factors are going to get together with the RNA polymerase. Now for the eukaryotic cell, because regulation is so critically important, it's going to be necessary to have these regulatory factors present, these basal transcription factors present before anything can happen. Sometimes it'll be a combination, sometimes one or two. It depends on who they are and where they are and how many are bound that will communicate to the RNA, whether polymerase, whether it's time to do the transcription, how fast to do the transcription, how much to do the transcription. So the main thing to take away from here is that you have this initial binding to the Tata box, recruitment of these other factors, transcription factors by letters here, and that's going to activate the RNA polymerase. And what you'll notice is here's the Tata box, and then if you follow the DNA off to the right here, you find that the active site of the RNA polymerase is really far to the right. And that is consistent with the previous slide that showed those the consensus sequences, and then way off to the right you saw that little green box that said this is where we start. All of these proteins bind to this DNA to perfectly space everything exactly correctly so that that initiation site is going to be perfectly placed in the active site of the RNA polymerase. And the RNA polymerase will recognize that and say, this is where I'm supposed to start, let me go. Now let's back out just a little bit more and let's look at some more elements here that are going to make this happen with more regulation. So here's where we've been before, right here. This is the DNA is all here. There's the gene, that's the part of the DNA that we want to turn into an RNA transcript that can go out and do the work of the cell. We've seen the promoter complex already. We've seen the Tata box, and that's where the Tata binding protein will bind and recruit all of those different factors, and the RNA polymerase will sit right here, and that's where it's going to transcribe. But the question is that sufficient? Do you just want those complexes and everything to bind to the promoter and just transcribe the gene over and over and over again? Or do you want to regulate how likely it is that this gene gets transcribed? And so even further upstream when you move in the 5' direction here, even further upstream, there's going to be regulatory elements here. These are going to be bits of the DNA that are going to contain information for other proteins to bind. They could be called activators, which means they make it easier for the DNA to be transcribed. They could be repressors, which means it makes it harder for the DNA to be transcribed, okay? Enhancing or repressing or activating, all the words sound like what they do. And these are called distal control elements because they're far away from the site of action over here. How do these things communicate together? Well, this is empty space here, or so it seems, but there are very specific DNA binding proteins that will bind onto this DNA, and they have shapes and they go through conformational changes that will bend the DNA, and it will bring this distal piece now proximal. It'll bring those two pieces together by making this little hairpin or this little loop structure there. And when these regulatory elements come close to the promoter complex, it becomes possible for these mediator proteins. These are those regulatory proteins. They say, hey, I'm supposed to bind here to this promoter complex and tell it it's good to go, but who's going to tell me to bind here? Well, the distal regulatory enhancer or whatever sites down here are going to bind to those mediator proteins. And when they all come together, then this becomes a functional, activated, ready-to-go complex. In this case, you're looking at an activator and you're looking at an enhancer. So when these things come together, it recruits all those transcription factors. It recruits the RNA polymerase and, of course, the sigma factor. The RNA polymerase is now present and it's time to go. So while the sigma factor in the RNA polymerase could bind to the top box and be in position, what this is doing is it's telling the RNA polymerase, I want to activate you, I want to enhance you, I want to make things easier for you so that you're more likely to make these transcripts because the cell needs your work as quickly as possible. Now let's look at how genes are organized in the DNA, because remember, we're making copies of the genes, and those genes are then turned into RNA, which is the working copy, and the RNA is what helps the cell do the work that needs to be done. Now for prokaryotes, with the DNA that they have here, remember there's not a lot of highly sophisticated DNA stuff. There is, of course there is, not like eukaryotes though. And so prokaryotes will put their genes all together in groups here. And this is called polycistronic mRNA, which means the genes, these different proteins or different components of the cell process or whatever that's needed, are all placed together. And a good example is going to be the tryptophan operon. Now the tryptophan operon is going to be a bunch of genes that are responsible for synthesizing the amino acid tryptophan. If a bacterium or a prokaryote is swimming around and it can get amino acids from its environment, you don't need to synthesize the tryptophan. But if you're in an area that's not very rich with nutrients, then you need to synthesize that tryptophan. And so these genes will then be transcribed. And so you start with the RNA polymerase, and the polymerase will move through all of these genes, and off will come one RNA transcript. One message will come off of this thing. And so this is called the tryptophan mRNA that has all of these, the message for all of these genes are on there. And where you have initiation sites for protein synthesis, we'll see in the next lecture, the ribosomes will bind and it will make protein number 1, protein number 2, protein number 3, protein number 4, and off they come and they can assemble and start making the tryptophan amino acid. Polycistronic, meaning multiple genes are contained within one mRNA. Now for eukaryotes are more regulated and they do a better job of mixing and matching. For the prokaryotes, they just say, you know what, I need tryptophan. If I need tryptophan, I want my proteins, and just give me my proteins, so let me make my stupid amino acid. Okay. But the eukaryote says, well, you know what, we may need a little bit more of 1, we may need more of 5, we may need more of 3, so over time, these different genes have gotten separated, and so there'll be distinct genes located at different places on the DNA within the chromosomal structure. And there are still going to be signals that say we need to make the amino acid tryptophan, and so we need to make the enzymes that will do the reactions that will help us make this. And so you can activate transcription at each one of these places, and they're going to be similar with their control elements, so that if you need tryptophan, you're going to turn on all these genes. But instead, what happens now is you get an mRNA for one protein, an mRNA for another protein, and so forth. And now this allows the eukaryote to say, now wait a minute, I have a lot of 5 and I have a lot of 1, but I don't have much 4 and I don't have much 3. And so it can turn on 4 and 3 to make more copies of those two because it has enough of the others. For the prokaryotes, they just say, give me everything, I need everything. But the eukaryotes can be a little bit more finely tuned, which is why having this separated makes a little bit of sense from a regulatory point of view. And you can take this out into the human genome and you can start thinking more widely about any particular gene that would be on there when you come together. So here is the DNA shown along here. And where we've been before is we've seen these control elements like the TATA box and the other things and the promoter, it's all there. There's going to be some initiation site that's going to tell the RNA polymerase to start and to do its work here. Over here are going to be the distal control elements that are far away. And remember that DNA bending proteins and stuff combined here and bring those together. Here's some closer. These are called proximal control sites that will modify how much this promoter, how active the promoter is and what happens there. Now there's one more thing to point out too that we really haven't said yet. There's two different types of DNA here. There's exons and there are introns. And they're sort of mixed and matched in there. And the exons now are going to be the part of the gene that you want to turn into the RNA. And then the introns are extra bits that you want to get rid of. And for a long time, the introns were just considered junk that had gotten inserted and it just needed to be replaced. But it turns out there's information over this entire DNA sequence here. The whole DNA has all this information about how to splice out or splicing or removing these introns. It contains information about how to bring these exon pieces together to let them be rejoined. There's information in there about structure, and it's a lot of information in there. So the introns aren't just junk. Yeah, they get cleaved out. Yeah, they get removed, but they are important. So in the process of making the RNA, you come up with what's called the primary RNA transcript. You get the whole thing here. And then you go about removing these intronic sequences here, and you're left with only the exons. Now there's two more things to tell you about here as well. For eukaryotic genes, there's going to be a thing called a 5' cap. And that's exactly what it is. It's going to be a phosphorylated guanine that's going to serve as a protector of the 5' end. It's going to be recognized. It's like putting a hat on, and that's exactly why it's called a cap. It identifies and protects that 5' end. And then over here on the 3' end, we have a thing called the polyadenylate tail here, which is this long repeating just A all the way down. And you're adding extra A bases here that's going to protect the 3' end from exonucleases. Once this messenger RNA gets out in the cell, it's going to be doing the work that it needs to do, but there are also enzymes out there that will work to degrade that messenger RNA. And so between the 5' cap that will protect this RNA and the poly-A tail, you can let this thing have a longer lifetime. Notice that the poly-A signal sequence is part of the original gene. That's the signal sequence we'll see in a couple slides that allow the synthesis of that poly-A tail, and you'll see there are enzymes that will come and do this cap for us as well. Okay. So here's the eukaryotic gene process. Of course, the DNA is here. You've now melted that DNA, and it's kind of a little bit weird how it's showing you here, but it's just showing you that one. But here is the messenger RNA that's coming off, in this case, I guess, the RNA transcript that's coming off. And you'll notice that right away, as soon as that little bit of RNA comes out here, there's going to be the synthesis of that 5' cap. And you'll see it coming up in a slide. And there's a polymerase II enzyme that's going to be very responsible for this here. And as this synthesis continues, as you make more and more and more of the RNA, here comes the pre-RNA or the pre-transcript, if you would. It's got this 5' cap because it was put on right away. There's the message, and then you're going to have this poly-A here, the poly-A tail. Now, if you recall, there's a bunch of stuff here you don't need. There's exonic and intronic sequences in there, and so you start this process called splicing, and we'll see that come up here in the slide, where bits and pieces of that RNA know to come together, and there will be factors that will bind, and they will catalyze the reaction of clipping the intron sequence and removing it and joining the remaining exonic sequences. And so now your processed RNA is smaller because you've removed those introns. You still have your 5' cap, you still have your poly-A tail. This thing is now recognized as a mature RNA. It can lead through the nuclear pore and go out in the cell and do the work that it's supposed to do here. What's left over is just going to be degraded. You'll get the nucleotides out of this, and they can be used for DNA and RNA synthesis and so forth. Here's the cap, a little bit more structure for you. You see this methylguanosine is this. It's methylated, which helps distinguish it from normal guanosine. It also has these 3 phosphates. As you recall, for most of the bases in this polynucleotide, you only have one single phosphate, and that one single phosphate is a phosphodiester here. But this has 3 phosphates, which is unusual. So this triphosphate linkage and the fact that you have a guanosine and the fact that you have a methyl group recognize this thing as a 5' cap. It's got a very distinct structure, and there are proteins in the cell that will recognize this and recognize that it's an okay structure, that the mRNA belongs in the cell and it should be there. Okay. It really does decrease the rate of degradation by protecting that messenger RNA. Now let's look at that splicing process, the removal of introns. And if you take this picture and multiply it by about 1,000, you'll see the complexity. This is a fascinating, fascinating area. And there's a lot of detail here, and I really don't want you to get lost in the detail, but I do want you to recognize some of the terms like spliceosome. And the spliceosome is going to be that group of enzymes and proteins that are responsible for doing the work of the splicing. And I want you to recognize that there are exonic parts and intronic parts of the messenger RNA, and recognize that there are a family of factors that do this. Okay, so what do we have here? This is the pre-mRNA. This is the primary transcript that has come off. And if you see here, there's going to be bits of the RNA that correspond to the exon of the gene. There's going to be a bit of this RNA that corresponds to an intron on the gene. And then over here, you've got an exonic sequence again. And the goal of the spliceosome is to know how to cut the intron out, get rid of those pieces, that piece of RNA, and then to join the two exons together. So it has to recognize the crossover space here between the exon and the intron, and on both ends. And it has to bring all these things, these two pieces together. How does it work? Well, the first thing you're going to have is you're going to have one factor that's going to recognize a very specific site inside that intron. It's going to sort of serve as a base. And you see here's U2 down there. It recognizes that. And then you're going to have another factor that recognizes this very, very specific sequence that represents the crossover from exonic to intronic sequence. And it's going to know to cut exactly at that base right there. That phosphodiester bond has to be cut. And so when these two things bind, they come together and they recruit other factors. And this U1 now is an enzyme that will cut the phosphodiester linkage between the intron and the exon, but it still holds on to those two pieces. You need to hold on to this bit of RNA because you have to connect it to the other exon. And the way to do that is you just take the bond you broke off of the intron, you take it off of the exon and just stick it on that A base there. And you're going to make what's called a lariat. This is what's called like a little loop or whatever. And that now has cleaved this bond. And the same thing is going to happen over here. This U5 is going to recognize very specific sequence between the end of the intron and the start of the exon. And the same process will happen. These two factors will fold over and bring the exon piece number 1 to exon piece number 2. There will be a new ligation reaction that will also break the bond between the intron and the exon. And so you've removed that intron piece, there it is, and then these factors will recognize that they're no longer part of the larger messenger RNA. They will come off and they'll just release this piece of RNA, which is called the lariat. And the last thing to talk about with the formation of the mature RNA, messenger RNA, is to talk about the poly-A tail. And there is going to be a consensus sequence in this messenger RNA here. Here's the cap, there's the message, and then towards the end here you're going to have this consensus sequence that will allow the machinery to recognize that you're nearing the end. And then here's going to be a cleavage site, and when you break that bit of the RNA off, the rest of the stuff is going to be trash. It's just going to go away. But this is going to be recognized by the machinery that will come in and say, my job is to add a poly-A tail. And so you bring ATP in, adenosine triphosphate, and the adenosine triphosphate will do the same reaction we've seen before. We have a nucleophilic attack, you get the release of pyrophosphate, but the adenosine with a monophosphate is going to be attached. So A, and it just goes on forever here. And again, you're just simply adding all of these A bases to make a long tail that will protect from exonuclease activity and preserve the lifetime of this message. All right, so now we've talked about the messenger RNA primarily, and that's what we've been talking about so far. But we also know that there's ribosomal RNA, and the ribosomes are going to be the machines that are going to help with protein synthesis. And there's also transfer RNA, and transfer RNA work with the ribosomes to help synthesize the protein, and the transfer RNAs are going to interact with the messenger RNA to let the ribosome know which amino acid comes next. So let's look at these different RNAs as well, ribosomal and tRNAs. And I really like this picture. I don't know, something about it is aesthetically pleasing. But for prokaryotes, when you have the ribosomal RNA and you have the tRNA, if you remember, the prokaryotic genome is set up so that you just have gene, 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 gene. It's very simple. And the same thing is going to be true for these different forms of RNA. When it comes to the part of the DNA that's responsible for making ribosomes and making tRNAs, they're going to be part of this long primary RNA transcript, just this one long piece, and it's going to come off as just a piece of RNA. Now, if you remember from the first nucleic acid lecture, RNA tends to be single-stranded, but it does have the ability to form double-stranded stretches here. And then it's very complex the way it works. And so as these things come out, they will form long stretches of double strand here. And then you've got this big loop and stretch and loop and so forth. And these little bits of pieces here that come out are very important structurally. They form and they recognition sites that tells the cell what to do with these. And you notice the tRNAs are going to do the same thing. It's part of the primary transcript, but we saw the tRNAs in that first lecture, and we know that it forms little bits of double-stranded RNA. It also makes loops and double-stranded RNA and loops, and that's important for its structure and its function. Over here is going to be the 5S RNA, which forms its own small structures, 23 and the 16, and these things are going to combine together. So these little arrows are just simply showing you where the RNA transcript is cleaved, and there's an enzyme called the RNase P, which is the enzyme that catalyzes the cleavage of these bits here. You just take off these bits of RNA. Here you're going to cleave the intact tRNA, and the intact tRNA comes off over there. Here the intact 5S RNA comes off. And the way these things are cut is going to change their final structure, it's going to change their final shape, and it's going to change their function. So in eukaryotes, it's very similar. Again, you've got this long stretch of DNA, and from those genes, you're going to be getting your RNA transcript is going to come off. And in this case, because we're talking about ribosomes, you're going to have the 45S ribosomal RNA precursor, and the precursor will come off. Now, for ribosomes, I didn't mention on the previous slide, but it's shown here nicely. Ribosomes are going to be composed of proteins, which are amino acid and polypeptides, and RNA. And so you have this large mass of proteins, you have a large mass of ribosomal RNA, and then you have these two pieces that can come together here to make the intact large subunit there. And again, like everything else, there's going to be some processing and some trimming and some modifications that change the overall structure and function and shape and so forth. And then it comes time to split these things apart. And so the first structure that comes together is going to be split. You have a small ribosome and you have a larger ribosome. Again, some additional processing takes place, and out goes the 60S subunit. If you recall, 60S just simply means what its size is. And then you've got the small one over here, the 40S. And these are two competent pieces that can work together to make a functional ribosome. They're exiting out here through these nuclear pores as we saw before. So what this is showing you is you have primary transcripts, and again you do the processing and the editing until you get these things down to the final form, and you've got the two different final forms of the RNA that's going to be used. There's a lot of gobbledygook here, but I think if you just understand the idea that ribosomal RNA and proteins come together and they're assembled and then processed and so forth, that's going to be good. What about tRNAs? Okay, we introduced the structure of the tRNA, and you see this very interesting shape here, where you've got a linear strand of RNA, but because of the nature of the base pairs, this thing will adopt some higher structure here, which is going to be base pairing, which allows it to make these arms here, and the arms have loops, and the loops have names, and there's very specific structural features. We'll see this coming up in the protein synthesis lecture next. Okay. But what's important now is that you've got the 3' end here, and at the 3' end, this is where the amino acid is going to be attached. A transfer RNA is going to transfer amino acids onto a growing polypeptide. And the other part of this molecule right off the bat is called the anticodon. And the anticodon is going to be that part of the tRNA molecule that will interact with the messenger RNA, and by forming specific base pairs, the messenger RNA will say, I want this amino acid. The base pairing will allow this one with a particular amino acid to come. We'll see that in the next lecture. And of course, the structure doesn't look like this. It looks like this over here, which is really fascinating. Here's the anticodon that allows this thing to bind. Of course, here's where the 3' end what the amino acid is, and we'll see better pictures of this coming up pretty soon. Let's look more closely at the tRNA synthesis here, especially in eukaryotes now. And you see there's the primary sequence here, there's going to be exons, there's going to be introns. But notice that there are promoter components here as well. And the promoter complex is usually, or component is usually all grouped together at one end. But in this case, the promoters are separated and they're within the sequences here of the exons. And that turns out to be very important. And the reason for that is that you need to do quite a bit of modification to this tRNA. There's very special bases in these loops that must be placed and then modified in order for this tRNA to have the exact correct structure for the exact correct function. And these promoter components are going to change the pace and the rate at which and the process by which this primary transcript is going to be processed to let all of these things happen together. There is an RNA polymerase that's specifically designed for this, that recognizes these promoters and these split promoters, that knows that it's time to produce this molecule in a very special way. So here's the primary transcript. It adopts that general cloverleaf structure that we've seen before. You're going to see splicing under the intron. And you'll see now addition of the specific bases that are needed there to finalize that loop. And then you've got, of course, the special bases being made. So here's a nice summary slide comparing eukaryotes to prokaryotes. And there's a lot of detail here, more than what we go into in this biochem class. It's a little bit more, but there are a couple messages to take here. And that is that prokaryotes are simple. Their DNA, basically one circular chromosome, one bit of DNA. There's no nucleus, and so this is out in the cytoplasm. And so all of the DNA synthesis and replication machinery, the RNA synthesis machinery is all in the cytoplasm, and it's all going to work together. And when the mRNA transcripts are being made, as soon as that mRNA transcript starts coming off of the RNA polymerase, immediately the transcription apparatus is going to start protein synthesis as well. And we'll see that in the next lecture here. Because bacteria, they need their proteins, and they're just going to be making their proteins as soon as that mRNA comes off. But eukaryotes, because of the complexity and the level of regulation and the mix and match nature that we have with our genes and our transcripts and stuff, we can do a little bit better of a job here. And so we're contained with our genetic material is contained within the nucleus, and that's where the DNA synthesis and replication is. That's where the RNA transcripts are made. Of course, there's mitochondrial DNA, and we'll talk about that. But we're talking about the nuclear DNA right now. There's a lot of DNA that needs to be operated on, and we'll talk about access and stuff during the regulation lecture. But the key thing is that when the mRNA comes out for eukaryotes, we have one mRNA per one gene, whereas the bacteria has many genes on one mRNA. And we have intronic sequences, and that's going to help us do these splicing reactions, and the splicing is what lets us have variation from one protein to the next. For the bacterium, what you see is what you get. The gene transcript is made, and it is used for transcription and translation right away. And then for our case, we're going to make a mature mRNA that's going to exit the nucleus and go out into the cytoplasm, and it gets picked up by the protein synthesis apparatus to start making proteins. Some key concepts, again, going through this and just making sure you understand these ideas can be a great way to pull it all together. So here's a question: when and where are you likely to find spliceosomes? Okay, so not a very complex question, but it's a great way to test a fundamental concept of this lecture. What is the spliceosome? Do you remember? And what is the job of the spliceosome? Well, are spliceosomes present in prokaryotes? Are they present in eukaryotes? And knowing that answer, where in that cell would you find the spliceosome? That's what you want to be thinking about. So it's asking if it's in the cytoplasm of eukaryotic cells or prokaryotic cells. It's asking, is it in the nucleus of eukaryotic cells during translation or during transcription, the cytoplasm of prokaryotic cells during translation? Well, if you remember, prokaryotic cells, really the message that comes off of the DNA is pretty much in its final form. There's no processing, there's nothing going on, and so you don't need to splice the RNA, you don't need to process the RNA. So any answer that has to do with prokaryotes can be ruled out right away. B and E are gone. And when is the RNA being spliced? Well, you have the primary transcript that comes out, and you have to take out the intronic sequences and bring those exons together, and whatever modification needs to be done. So it sounds like the primary transcript is being modified to make the mature transcript that actually leaves the nucleus. So it sounds to me like it has to do with the nucleus and something to do with transcription. So where do you think you're gonna find it then? Well, prokaryotes out, prokaryotes out. Transcription in the nucleus of eukaryotic cells during transcription. Yeah, sounds pretty good to me. In fact, that's what it is. This is the spliceosome complex here, and of course this shows you that you have the primary mRNA transcript that's now being processed into the mature final form that's going to leave the nucleus. Which of the following takes place first during transcription of a gene in eukaryotic cells? Wow, well, you could think about anything. You could think about promoter complexes or factors or DNA bends, or you could think about anything. So in this case, you kind of need to look at the answers to figure out what the possibilities are. And so you see these are very short answers, and so you can move through them pretty quickly. Poly-A addition, intron removal, 5', all these different things, okay? So what do we know? We know poly-A tail is going to be done pretty much at the end. That's when you're trying to elongate the chain. The intron is going to be part of the splicing. That's when the primary transcript is made. 5' capping. Oh gosh, yeah, I remember. As soon as that RNA transcript starts to come off of the RNA polymerase, you want to protect that thing. And that 5' cap gets stuck on there almost immediately. So that sounds like a good one. And then how about the transcript binding to the 18? Well, that sounds like the transcript is already out in the cytoplasm interacting with the ribosomes. Or here, the transcript export from the nucleus. That sounds like the mature transcript is ready to go. So these sounds like late things to me. So it sounds like maybe answer C would be the one. Sure enough, there it is. You see the 5' cap gets put on as soon as that little bit of RNA comes off. And that helps protect this thing. Thank you.